Hello students, this is Dr. Faizan Mirza. I'm covering the details of how the RNA polymerase functions in producing the transcript of mRNA. So this is the RNA polymerase enzyme. When the RNA polymerase enzyme is allowing the mRNA to be produced using the template strand of the DNA, we know that there are two strands of DNA. One is complementary to the other. So the template strand behaves in such a way that the mRNA transcript is copied, or you can say made by complementary base pairing, using the exposed bases here and the free ribonucleotides of the RNA, which are then joined according to complementary base pairing. So in this case here, the diagram, you can see this thing again, that when the RNA polymerase is joining these free nucleotides, again they are joined in the same direction, 5 prime to 3 prime. So the growing chain of mRNA molecule will be 5 prime to 3 prime, because this is the direction in which the enzymes function. So this is how it keeps joining, which means that the region which was produced earlier in the growing mRNA molecule will have a 5 prime end here, and towards the end it will have a 3 prime end. And when you have a 5 prime to 3 prime direction, in this case, the RNA transcript is actually being made using a template strand, which in turn is having 3 prime end opposite in that end, and the 5 prime end at the opposite end, because we know that they all work in the opposite directions. So the direction of copying will be this, that as the bases get exposed on the template strand, new nucleotides will keep on getting laid down in the mRNA transcript. When the mRNA is being made, you know that your DNA is a very long molecule. There will be a start triplet or a start codon. The start codon is located on the template strand, which is 3 prime to 5 prime. This template strand is having the start codon. The start codon will always be TAC. TAC codes for the amino acid methionine. So this TAC will be the first amino acid to be placed, and as the codons they get transcripted into mRNA. Eventually, you have a stop codon. The stop codon or the stop triplet is stop triplet ATC or ATT or ACT. Either of these three will be located here, and again they are present on the same 3 prime to 5 prime template strand. When the stop codon comes, the mRNA transcript will stop being added further anymore. So new nucleotide will not be placed, and the mRNA will be free to move out of this DNA molecule. The exposed bases of the template strand and the non-coding strand of the DNA will start to coil up once again into a double helix structure using complementary base pairing and hydrogen bonds. Now, another thing which is of importance here, although beyond the scope of the syllabus, is that why always methionine is the first amino acid to be placed, because once the mRNA is made, you know that the first codon here will be TAC. So if this first codon here is TAC, we know that this TAC will be coding for the amino acid MET, and after MET, for example, the next codon codes for the amino acid lysine, and for example, after that you have leucine, and then you have proline, and so on and so forth. So what are you adding? You are adding amino acids in this direction. The first amino acid to be placed, methionine, will have this end blocked. So the amino acid cannot be joined in this direction, which means that a bond, you can say the bond which is made, which is the peptide bond. The peptide bond can only be made in methionine in the direction in which the next amino acid can be laid down is at this end. So this is the only end in which the methionine will allow the amino acid to be joined. Its opposite end, which can also otherwise participate in a peptide bond formation, will be blocked. And a methionine is the only amino acid in which your cells can block one end to make sure that the peptide bond gets made in one direction only. Let's suppose after pro you have another MET amino acid here. Now if the MET amino acid is here and again you have another amino acid pro here, so this MET amino acid will not exactly be as the first MET amino acid, although they both are the same amino acid, but this MET amino acid will have both ends exposed, at which a peptide bond can be formed at this point as well, and a peptide bond can be made at this point as well. Whereas the first MET which was made here, this was an amino acid in which one end was blocked, so the peptide bond could be formed only at one end. We know that every amino acid is having a carboxyl end and amine end as well. So if carboxyl end and amine end both are available, the amino acid can form peptide bond at both ends. So at the first MET, the one end is blocked to prevent the bond formation in the opposite direction. Although this entire description that I've just discussed, what the difference between the first MET and the MET coming anywhere else, and why MAT is always the first amino acid, translated into a polypeptide when the mRNA is being translated. This is actually beyond the scope of the syllabus, but it's worth discussing. Another thing that I'll be discussing here is the post-transcriptional modification of an mRNA in a eukaryote. We know that this is something interesting and something new which is added in the syllabus. So in the eukaryotes, the mRNA undergoes post-transcriptional modification. So what I'm going to discuss here occurs in eukaryotes only, not in prokaryotes. So when the mRNA is made in a eukaryotic cell, we know it's basically a transcript of the template strand of the DNA, a segment of DNA. But this mRNA will have sequences which are called introns as well as exons. So there will be introns, there will be exons, some introns, and then again introns, exons. So what are introns and what are exons? This basically is your mRNA in your eukaryotic cell, and this is called as the primary transcript mRNA. Now the introns and exons, basically they all not code for the amino acids. The introns are just there, they do not code for amino acids. So basically only the exons are the ones that will be translated into amino acids. So the introns must be spliced, they must be removed, they must be cut, they must be excised, and they should be cut by the enzymes endonucleases present inside the cell. So using the enzymes endonucleases, your eukaryotic cell just chops up this mRNA, it removes the introns, and only the exons are then joined together to make the final mRNA. So you can see that the introns have been removed from here, and this is the final mRNA which is produced. And you can see the if you just compare the strength, the length of this mRNA, we see that this length is basically way shorter for this mRNA as it was for the older condition. So this was a larger mRNA, this is a smaller mRNA, and this smaller mRNA is there because it's having exons only, and it is these exons that will be translated into amino acids. So for every codon in the exon, you will have an amino acid which will be there brought by the tRNA on the ribosome once the translation is occurring. Here again I've mentioned the same thing. For intron there will be no amino acid, for exon there will be amino acid. Again introns no amino acid, for exons there will be a chain of amino acids. So why keep the introns eventually? So the introns will be just removed during post-transcriptional modification, and eventually you will have a very short mRNA with all the unnecessary these nucleotide sequences removed, and this entire occurs before translation can proceed. So, and this post-transcription modification of converting the primary transcript of mRNA into mature mRNA, this is occurring in eukaryotes only. This is a feature of a eukaryotic cell, and this doesn't occur in prokaryotes. That's it from my side. Thank you so much.